Hello and welcome to our video on tumor suppressor genes. Somatic mutations can occur in any of the cells of the body except the germ cells, sperm and egg, and therefore are not passed on to children. The traits of cancer cells are consequences of genetic changes in the tumor cells, genomic instability, self-sufficient proliferation of growth, refractory to inhibitory signals, survival without survival signals, unlimited replicative potential, recruitment of blood supply, invasion and metastasis. Cancer cells contain multiple alterations in the number and structure of genes and chromosomes, mainly acquired by mutations in somatic cells. Two classes of genes affected by genetic and epigenetic alterations in cancer cells are oncogenes and tumor suppressor genes. Oncogenes contribute to tumor development by increased or misdirected activity. Tumor suppressors, insufficient or lost function, supports tumor development. A tumor suppressor gene encodes a protein that acts to regulate cell division, keeping it in check. When a tumor suppressor gene is inactivated by a mutation, the protein it encodes is not produced or does not function properly, and as a result, uncontrolled cell division may occur. Tumor suppressor genes represent the opposite side of cell growth control, normally acting to inhibit cell proliferation and tumor development. Tumor suppressor genes stop a cell from multiplying when DNA damage is detected. Specific tumor suppressor genes can trigger cell suicide in damaged cells. When tumor suppressor genes don't work properly, cells with DNA damage divide and can accumulate more damage, leading to cancer. Tumor suppressor proteins regulate orderly cell growth and differentiation by sensing the surrounding environment, transmitting signals to the nucleus and directly affecting transcription, translation, survival, or cell division. Repression of genes that are essential for the continuing of the cell cycle, effectively inhibiting cell division. Coupling the cell cycle to DNA damage, cells should not divide with damaged DNA. The cell cycle continues once it is repaired. If the damage cannot be repaired, the cell should initiate apoptosis, programmed cell death, to remove the threat it poses for the greater good of the organism. Metastasis suppressors prevent tumor cells from dispersing, block loss of contact inhibition, and inhibit metastasis. DNA repair proteins are usually classified as tumor suppressors: HNPCC, MEN1, and BRCA. The two-hit hypothesis given by A.G. Knudsen provided a unifying model for understanding cancer that occurs in individuals who carry a susceptibility gene, and cancers that develop because of randomly induced mutations in otherwise normal genes. The two-hit hypothesis provided a unifying model for understanding cancer that occurs in individuals who carry a susceptibility gene, and cancers that develop because of randomly induced mutations in otherwise normal genes. Cancer requires both alleles of a gene. If only one gene allele is damaged, the other can still produce the correct protein. When a tumor suppressor gene is inactivated by a mutation, the protein it encodes is not produced or does not function properly, and as a result, uncontrolled cell division may occur. Such mutations may contribute to the development of a cancer. Transcribing and translating tumor suppressor genes creates protein signals. DNA code mutations can disrupt tumor suppressor genes. Changing a tumor suppressor gene's DNA code prevents signaling. One missing letter in the mutated DNA message confuses the cell. The cell may not become cancerous. A mutation is a change in the DNA sequence of an organism. Mutations can result in errors in DNA replication during cell division. Mutations are gene defects. They are abnormal changes in the DNA of a gene. Mutations involve changes in the arrangement of the bases that make up a gene. Even a change in just one base among the thousands of bases that make up a gene can have a major effect. A mutation can affect the cell in many ways. Some mutations stop a protein from being made at all. Some mutations may cause a gene to be turned on and make more of the protein than usual. Some mutations don't have a noticeable effect, but others may lead to a disease. Most oncogenes are mutations of certain normal genes called proto-oncogenes. Proto-oncogenes are the good genes that normally control what kind of cell it is and how often it divides. When a proto-oncogene mutates, changes into an oncogene, it becomes a bad gene that can become permanently turned on or activated when it is not supposed to be. When this happens, the cell grows out of control, which can lead to cancer. Oncogenes are mutated forms of genes that cause normal cells to grow out of control. A cell continuously receives messages, both from its own genes and from other cells. Some messages tell it to grow, and some tell it to stop and rest, like the accelerator and brake pedals in a car. Tumor suppressor genes act as a cell's brakes. They encode proteins that restrain cell growth and prevent cells from becoming malignant. Oncogenes act as accelerators of cell proliferation, encode proteins that promote the loss of growth control and the conversion of a cell to a malignant state. The go signal keeps the cell dividing and in the cell cycle. The stop signal stops dividing and exits the cell cycle. These stop and go signals help maintain healthy, functioning cells. Tumor suppressor genes code for proteins that tell a cell to stop dividing. Proto-oncogenes code for go signals that tell cells to divide. Built into the cell's machinery are two signals: the go signal to stay in the cell cycle and keep dividing, and the stop signal to stop dividing and exit the cell cycle. These stop and go signals work to maintain the correct balance of healthy, functioning cells in the body. Well-described examples of tumor suppressor proteins include Rb, p53, APC. Rb, the retinoblastoma gene, is the first tumor suppressor protein discovered, was the first retinoblastoma protein, PRB, in human retinoblastoma. Another important tumor suppressor is the p53 tumor suppressor protein, which is the guardian of the genome encoded by the TP53 gene, also known as protein 53 or tumor protein 53. p53 was identified in 1979 by Lionel Crawford. It is one of the most frequently altered genes in cancer which monitors DNA damage. When DNA in a cell is too damaged, p53 normally sends a signal that tells the cell to commit suicide. The cell can monitor itself for DNA damage during the cell cycle. If DNA damage is detected, the p53 protein plays an important role in putting a brake on the cell cycle. The p53 protein prevents the cell from completing the cell cycle if its DNA is damaged or the cell has suffered other types of damage. When the damage is minor, p53 halts the cell cycle, hence cell division, until the damage is repaired. The damage is major and cannot be repaired. p53 triggers the cell to commit suicide by apoptosis. These functions make p53 a key player in protecting us against cancer. That is, it is an important tumor suppressor gene. Tumor suppressor genes function as guardians of our cells by preventing cells with DNA damage from dividing and passing on harmful mutations to daughter cells. Some tumor suppressor proteins function as a braking signal that stops the cell cycle when a cell with DNA damage is detected and needs to be repaired. Other tumor suppressor proteins instruct a cell with damaged DNA to commit cell suicide. Thank you for watching and have a great day.